Welcome everyone to today's breakfast seminar. We are very happy that you decided to join us today in the morning. We have exciting seminar ahead. I would like to start with agenda of today's meeting. And in case you are still looking for the place to sit, there is plenty seats in front. So let me start with a short introduction of what you could expect today. Firstly, Oliver Riener, head of Biognosys Group, will give you a short introduction and also company announcement. And after that, we will have 3 external speakers. First would be Jochen Schwenk from KTH Royal Institute of Technology, talking about circulating biomarkers of treatment response, and this will be insight from the pilot study enhanced by mass spectrometry. Afterwards, we have Maria Valle from Max Planck Institute of Biochemistry, and she will be talking about high sensitivity immunopeptidomics and how this approach helps with discovery of kinase-regulated tumor antigens in CML. And the last speaker of today is Daniel Grimm from Grey Wolf Therapeutics. Daniel will also focus on immunopeptidomics approach that help with the project where IRAP1 inhibition alters landscape of immunopeptidome and also abrogates presentation of putative APAXA autoantigens. And I would like to invite first speaker, Oliver. Please, let's start today's meeting.
Good morning, everybody. So we have 3 exciting speakers upcoming. So I just give you 3 very short announcements. Everything else we can discuss at the booth. So first, Biognosys Group. So Biognosys, PreOmics, Biocrates, and the PepSep are coming together to form the Biognosys Group, an affiliate of Bruker. So our mission is really to enable mass spectrometry across all steps of the workflow, starting from sample preparation, quant kits, columns, software, and the most direct access to technology, CRO services. We are vendor neutral, and we believe that the field needs both manual and automated solution, and this is how we develop our products. And the first product that's already a result of this union is our P2 IST plasma that brings the best of 2 worlds together, Biognosys P2 single particle enrichment system and PreOmics very well established robust IST technology. So it's a solution that is accessible as a kit for manual use and for automation. So our focus is always to use customers' existing automation. So you don't need to buy a new robot just for this one workflow. And we help you bringing this into your systems. And our second launch, our first software launch, new software product launch since quite some time, is Omics Flow. So one of the themes that you see here across the conference is ever bigger data sets, deeper data sets, immunopeptidomics, searches with multiple PTMs like you can do now with the Spectronaut 20 that we launched at the ASMS. And Omics Flow brings Spectronaut into your cloud, so into your existing clouds of companies, but also academic users. So it's optimized to run Spectronaut in the most efficient way, using nodes and spinning them up in a way so that Spectronaut gets really scalable. Come to our booth, talk to us, and we can show you, and we're very happy to show you all our products together. And with that, I am handing over to Jochen. I'm looking very much forward to hearing your talk. Thanks.
Okay. Thank you so much, Oliver, and thank you all for coming up this morning, especially the people coming in from the West Coast. My name is Jochen Schwenk. I'm working out of Stockholm at the KTH Royal Institute of Technology and Scilab Lab. And many of you maybe have seen me in sort of my different roles here at UPO. Most importantly, I have the great honor, pleasure, and joy to work with a fantastic group out of the community in the Human Plasma Proteome project. And many of those that you see here are also present at the conference, so it's really a fantastic environment. So if you're curious what we're doing, every second year or so we're writing a new review. We are currently in the process of preparing the next one. But if you're curious and want to engage, please reach out to anyone that you see on this list, where we have really the idea to bring together activities from the community. We also touch upon different technologies, and maybe some of you would regard me as working with the Human Protein Atlas and an antibody guy, but I'm really curious about technology, and I think the fantastic opportunities that have arisen in our community, and Biognosys being one of them, of course, makes us curious to try these technologies. And I had really the great pleasure to get an email from Lucas a couple of years ago asking to connect and discuss some ideas. So it's also in his name that I'm here, and I'm thankful to his connecting us. It's also a funny event that Oliver and I have been studying together many years ago. So our last experiment was probably in organic chemistry and trying to fill Oliver's specialty, fill small vials from large bottles and being successful at some point in time. But also, I mean, that you experience how somebody you have worked with as a student or studied together as a student has really achieved a great impact in the community. So also congratulations at this point. But of course, when we work with a community that has been, I think, tremendously growing over the last couple of years, matured in its way of development techniques, and maybe some of you who have been at UPO many years ago looked at many chromatograms, many spectra, and I think nowadays you actually look at how this data comes into action. But of course, we are reaching a new scale where UK Biobank and other activities have really sort of enabled not only us, but the greater community out there to work with proteomics. But of course, we really need to make ourselves more sort of reliable in terms of that everyone can use proteomics as a big sort of theme in their research, because the information lies in the proteins, as we know. So we have come together with a couple of people that around Tianan, Uwe, and Jenny to discuss how can we standardize plasma proteomics in a way that is, like say, vendor neutral, technology neutral, or agnostic, so we can actually bring together data before the data is generated. So we have everyone has the opportunity to get a standardized sample in the future that you can run when you have an optimized workflow, benchmark it against existing data. So that's, I think, also an important aspect. And I think we also are seeing a new horizon. I'm just going briefly through this because I think from a technology-driven community, we really want to help and improve lives. And I think that's where I think proteomics can really play a big role. As I mentioned, we can do it in a large scale. We can do it high throughput. Thanks to all your work, we can measure thousands of proteins with high reliability, sensitivity. But now what we do with all that information, and I think here really we are at a critical point as a community to take the data with great care. Because there are so many things that influence the phenotypes we are measuring. And I think today we want to present these two examples of how medication would impact your proteome and the effect of the treatment that it can have on the life of a person. Of course, that's very important. And we have different aspects, of course, when we take a sample, because that will, again, define the phenotype that you're measuring. So we think about the way that we are approaching science today. I guess we're still in the middle of this picture. So we're looking at a molecular phenotype. It's primarily driven by the technology that we apply, and it's driven by the sample that we have from the clinicians, or we may be collecting it ourselves. But I think where we really want to go is either to the left, so that's on the left, to finding the origin of the disease and actually treating the disease and monitoring the effect of the treatment. So we are sure that the molecular sort of changes that had occurred during the disease have actually sort of reverted. And the ideal scenario is, of course, to do intervention before the disease actually sort of manifests. But I think what we are still mostly sort of challenged by is that everything we are measuring is a systems biology picture, because proteins may be expressed by different organs, and they may tell us something about the comorbidities that are not maybe the center of our interest in a specific study. So it's, again, sort of a shout out to all of you to be careful, thoughtful when you interpret your data, and that you sort of learn from these community efforts that have already been out there. I think a key point is actually when we take a sample, because we often assume that what we measure is a static sort of phenotype, but we know that the proteome is changing over time. And I sometimes call this sampling at the speed of the proteome, because as we know, when you go on vacation, where you're super stressed about getting everything organized, running to the airport, packing up stuff, checking if you have your passport, your ticket, and what have you. And of course, maybe you have to send the last email to your colleagues so you can sort of try to switch off. And your proteome will look different than after you've come back on vacation 4 weeks at the Maldives, been diving, drinking some coconuts, water. So, of course, that will have an impact on who you are. And I think that we should keep in mind. So we need to, I think, really put our forces into longitudinal monitoring, because that will give us the certainty about the phenotypes we're measuring and that they're actually related to the event that we want to study. So, of course, as you know, breast cancer is really a global problem, right? It's the #1 leading cause of death in women in the Western world. And of course, it has a huge sort of impact on society. But of course, we've also seen how tremendously advanced our technologies are these days to actually diagnose. But the problem is not that we can diagnose women with high precision, and I think we have more and more better tools to treat them. But the problem is that it's often very late, because the tumor is immunologically cold. It's very hard to reach. And it doesn't really signal a lot of things out there that we can measure. So the ambition of the people that I had the honor working with for the last decade or so, the KARMA project in Stockholm, they have come together and sampled more than 70,000 women in Sweden who undergo mammography screening to understand what is the population-based sort of scale impact of breast cancer on society and how can we prevent breast cancer from occurring. That's the major mission. It's not to improve diagnosis. And one of the things that I've been focusing on is breast density, so the stiffness of the tissue. You can see an image here where high density, especially in premenopausal women, is a high risk factor for developing breast cancer later in life. So one ambition that they have is, can we find tools to modulate breast density as one of the key risk factors? And they have started a couple of years ago a preventive trial. So they've taken some women out of these 70,000 to enroll in a study that they call CHARISMA. And in CHARISMA, they have given, and I'm happy to show an organic structure here, so I'm very proud as a sort of proteomics guy to show a metabolite, that they have given women who have not been diagnosed with breast cancer. They came from different tiers of the risk factors that existed with a certain different levels of tamoxifen. And the ambition and the conceptual framework was because they observed in several studies that women who actually have received tamoxifen due to treatment got a reduction in the density of the breast. So the ambition is that can we use tamoxifen as a preventive measure to reduce the density of the breast and thereby reduce the risk of developing breast cancer? And of course, that is a quite sort of ambitious task. But the problem, of course, is when you have a molecule like tamoxifen, which gets metabolized to endoxifen, and which acts on not only the tissue it is intended to be acting on, it acts on the whole body. So we have run a clinical trial with them, taking 44 women who were in the placebo arm and 44 women in the 20 milligram tamoxifen arm. And the aim was, can we see any changes occurring over time, and can we see any changes related to the placebo arm changes? Because what we expect, and that's a little bit of a picture on the right-hand side, is that the drug will act on the estrogen receptor, and the estrogen receptor is not only found in the breast tissue, it's found in muscle and various types of tissues, so it has quite substantial side effects. So the problem really is that you cannot just give somebody a treatment, but you make the life of that person worse, right? So the preventive treatment should actually be an improvement or a bearable burden for the person, so that the effect of the treatment actually is of a greater benefit. So we had a great pleasure to work with Yuan and the Biognosys team to explore the possibility of using mass spec in order to understand, can we see any structural remodeling, because that's what people understand breast density is, and can we see any proteins changing in relation to tamoxifen use. So in this small study, we have measured about 4,500 proteins that we could quantify in all samples. And we've also have a study that we've done with Olink before, so we know that there's a complement of about 400 proteins in common and about 1,400 that we add on top of the things that we already have looked at. So it's really important for us that we can expand the dimension of this investigation, because it's not about us comparing technologies, saying one is better than the other. We want to find information that is actually of benefit for the patients and that we can use to better understand the effect and consequences of tamoxifen use. So now this one stopped. Here we go. But of course, when you have such a clinical trial, so you have a baseline before treatment and 6 months after, you first want to understand, does participating in the study have an effect? So here we look only at the treatment arm, and we see that during the course of these 6 months, there are not really very strong changes. We have some fluctuations that we expect because sometimes engaging people into a clinical trial will change their lifestyle. And it's the same for mass spec or Olink. So it's independent of the methodology. We may have some small indications here, but these are probably something that we can ignore once we move on and change another set of samples. But of course, we wanted to see, is there an effect of treatment on these participants? And of course, as you see here, it's substantially stronger impact on the circulating proteome 6 months versus the baseline measurement. So we know that, of course, treatment has an effect. And of course, now maybe the initial thought is, wow, this is amazing. We see a lot of things happening, and that's a treatment effect. But of course, as I mentioned, tamoxifen has substantial side effects, so we don't know yet whether these features that we see is a consequence of side effect or is actually a consequence of density. So putting these two things together, so we know that participation has little effect and treatment has some effect. So here we adjust for participation in the treatment effect. So we're actually looking at the so-called delta of delta. So we're looking at the differences between the changes occurring in the treatment versus the placebo arm. And I guess that's common practice in pharma industry, but I guess for many of us in sort of the academic universe, I think this is really the currency that is most important for us to use because it takes the variance of both of these changes into account. And we're quite surprising to see that we have effects on both technologies that we use, right? And you may argue, well, you have some that is more significant than the other. But I think at the end of the day, that's not really the point. I think the point is to identify features that will tell us something about the biology, and then we can go back, look at genetic variants, we look at previous health history of these women to better understand what are these changes that we're seeing. So, of course, how can you compress all these 4,500 data points into some sort of informative set of values? And I think one of the interesting things here, and that's work done by Paulina from the Biognosys, is to compress the data using different network models. So here we use WGCNA. And to our surprise, without asking too many questions and informing the system about too many changes, we actually see that one of those modules composed about 120 proteins was highly associated to collagen and extracellular matrix restructuring, which is, of course, something we expect. And many of these proteins that were in the model actually correlate to the different metabolites related to tamoxifen, and really key features that we think have a high value in terms of the metabolism, but also in the effect of the treatment. So which are these proteins? Here I show you some, and these are also going to be shown later today on a poster. This would be some of the proteins that give you information about the treatment effect on the liver, but they also give you some information about the effect of tissue remodeling. And I think that's exactly where we want to start. And we want to look at other proteins in relation to the collagen structure that are really important for us to really match these measurement data to the expected treatment response on a tissue level. So one may ask, yeah, well, why don't you detect low abundant proteins? And isn't that the purpose of doing these things? I think the purpose is to get the best possible data and to let the data speak for itself and tell you what are the information sets that are out there in relation to the phenotypes and the women that you have enrolled in the study. So we're quite excited, and we look at the tissues that some of these healthy women have allowed us to take or allowed the collaborators to take. We can now go in and actually do tissue staining and other things with this type of information, so we can measure the remodeling effects pre and post treatment. And with that, I'm at the end. I would like to thank the team at Biognosys, of course, the fantastic collaborators from the Karma project, everyone in my group, specifically Leo, who's been working very closely with the team at Biognosys, and everyone at the Protein Atlas, and for you showing up so early. Thank you.
Thank you, Jochen. Please, if you have any questions, let's keep them for the last part of today's meeting. We will have a common Q&A at the end. And now I would like to invite our second speaker, external speaker, Maria.
Yes, good morning, everyone. I'm very happy to be here and present one of my projects to you, which is about immunopeptidomics in kinase-regulated cell lines. Yes. So the idea of this project was to understand how kinase treatment modulates the immunopeptidome, in this case of CML cell lines. And for this, we used pharmacological inhibitors that are already released in the market and wanted to understand what's the profound impact on what is presented on the surface of these cells to eventually build a framework where we can use where we can use compounds that are released to the market for other purposes in the context of cancer here and to improve therapy outcomes and characterize the crosstalk. So this was the setup. We had a CML cell line. We used 3 different released kinase modulators you see here on the screen, and we compared it to our control cell line. The experimental design was as that. We had the cell line, the different treatment options, and we performed a multi-proteomics study here, where we used proteomics, phosphoproteomics, immunopeptidomics, and phosphoimmunopeptidomics to really understand the impact of these 3 modulators on the immunopeptidome, classified into 3 different mechanisms that we discovered here, which I will go into detail later. For the analysis of these data sets, we used Spectronaut 20 for the phosphoproteomics workflow, the immunopeptidomics workflow, as well as the phosphoimmunopeptidomics workflow. For those of you who have worked with Spectronaut 20 already, you'll see that there is a dedicated immunopeptidomics pipeline in there, which we used for the immunopeptidomics data set, and the phosphoimmunopeptidomics was searched with that pipeline, but we added phospho as a potential modification here. We performed the IMBAS-MS-based sample preparation, where we used these cell lines and enriched based on a biotinylated antibody fished with streptavidin magnetic beads in a more automated fashion. Then we separate the immunopeptides from the proteins via molecular weight filtration, and then we perform MS-based immunopeptidomics analysis here with the EvoSep and the Whisper-40 gradient on an ion optics column coupled to an Ultra-2, timsTOF Ultra-2 instrument using optimal DIA passive methods. So what we see first here is that we were able to achieve a deep and diverse immunopeptidome. We used around 5 to 10 million cells for this approach for all the 3 treatment conditions as well as the control. So there was no overall impact on the depth overall, but we saw quite some substantial regulation and quite some remodeling depending on the treatment and the treatment arm that we subjected it to. We got a diverse set of immunopeptides covering all the expected HLA protein types here. But what really struck us was the remodeling, that it was so profound, although the immunopeptidome depth was so similar that we were really able to clearly distinguish the 4 treatment arms here. Here you can see the modulated immunopeptides. So these, in the plot, you see the actual immunopeptides, not the phosphoimmunopeptides, but next to that, we also saw quite some significant modulation of phosphoimmunopeptides. And this is, again, the strategy that we used for comparing and setting this into context. We took these immunopeptides, the phosphoimmunopeptides, and now we compare it to how the phosphopeptides and the proteome from the proteomics experiment were remodeled to actually bring this all together. These are the 3 mechanisms I was talking about previously. So one of the mechanisms is that the kinase is directly phosphorylating the source protein, and this phosphorylated source protein snippet is directly presented on the HLA protein. The second mechanism you see here is that the kinase has an impact on the source protein stability. So either by stabilizing it or destabilizing it, but not the phosphoimmunopeptide that is presented, so more of an indirect mechanism. And the third mechanism is that the kinase has an impact on source protein production, and that the production or activation of transcription factors and increased production of the source protein leads to a change in antigen presentation. I've brought 2 examples out of this publication with me here. Here's an example for mechanism 1 and how we identified it in this context here. So here we use the senior framework. This is done by our collaborators for this project to really elucidate the connection and look for the shortest path between the kinase inhibitor that we used, the kinase, and the modulated phospholipid in question to correlate and compare kinase activity and directly relate it to the phosphoimmunopeptide presentation. And here is an example of a source protein. So in this case, the beta-ABL inhibitor, which is modulating the stability of CDK1/2 via this phosphorylation site. And here down in the middle plot, can't see it. You have the phosphoproteome impact, and then on the right plot, you see the impact on the phosphoimmunopeptidome. So the middle plot was drawn from this phosphoproteome and proteome data set, and the right plot you really see how the inhibition of this kinase has a profound impact on the presentation of this actual phosphoimmunopeptide, in this case from SCML2. The second example I've brought you here is the second mechanism where the kinase inhibitor has an impact on source protein stability. It's a similar principle. We're looking for the shortest way of how the kinase modulator might impact downstream through a lot of pathways the presentation of the source protein through looking into phosphoproteomics, proteomics, and immunopeptidomics data. Here's the second example. This, again, is now the Janus kinase, where we see, again, not so much of an impact on the phosphoproteome level, but again, when we look into the specific inhibitor, we see quite a reducibility for the vimentin phosphoside. But in this case, it's not the phosphorylated peptide that's presented, but the unphosphorylated peptide. So this is more of an indirect way of protein stability we're looking into here. And then the next step, we compared our data set to some published studies where they took patient-derived immunopeptidome data sets and compared it to benign immunopeptidome data sets to really see if we find common features that we'd identify from our cell line-based data set and could potentially translate to a clinical application here, if this is a valid model to analyze and to work with here. And indeed, we saw that we share 10% of the tumor-associated antigens from our study with these CML data sets that were not found in the benign data sets. So these 10% modulated antigens are modulated ones, not overall. And we see that more than 40 tumor-associated antigens are altered in the presence of our 3 kinase inhibitors. So this really shows us that there is potential avenue of using off-label drug in the context of cancer and modulating what is presented, hopefully specifically. So we used our benign immunopeptidome data sets here as a reference, but this has to be proven further in the future if this is a viable strategy to actually modulate through kinase treatment the immunopeptidome to make the cells more accessible for immunotherapy later. So with this, I'm at the end of my presentation. We showed that the pharmacological inhibition of selected kinases can modulate the immunopeptidome in this CML cell line. We showed a way of multi-omic integration, how we can really combine a very rich source of information by just using a mass spectrometer in this case, but still looking into very much different layers of proteome and immunopeptidome regulation to really elucidate kinase-dependent immunopeptidome presentation, and how the potential of combining kinase inhibitor treatment and immunotherapies might be a chance to enhance antigen visibility and improve, hopefully, antileukemic immunity. With this, I'm at the end of my presentation, and I want to thank Matthias, my supervisor, as well as Francesca, who spearhead these publication. This can be found on bioarchive now and is submitted for publication. I also want to highlight the other 3 main authors of this publication. You see there, Veronica, Georgia, and Valeria. Thank you.
Thank you, Maria. And as I said, please keep your questions for the last part, and I would like to invite our third speaker, Dan Green.
Good morning, everyone, and thanks to Biognosys for having me to speak, and I think also apologies to Monica for the complicated talk title. It's not an envious thing to read out. So the next 15 minutes, I'm going to walk through really how we an approach to modulate the immunopeptidome using this thing called ERAP1 inhibition, and how we've shown that this plays out as a therapeutic in an autoimmune condition called axial spondyloarthritis, and particularly these patients, these axial spondyloarthritis patients that have this HLA-B 2705. Oh, sorry, that was my title. So a little bit of background on Grey Wolf, just before I get started. So we are a clinical stage biotech company, and our scientific approach really is to target this signal 1 component of T-cell activation. So T-cell activation requiring a chain of 3 signals, where most current therapeutic approaches are focused on the latter stages in this signaling cascade. But what we've really shown is that by manipulating signal 1, which is antigen presentation, we can impact the visibility of cells to the immune system. And this plays out in oncology, but it also plays out in autoimmunity as well. So ERAP1, which is the enzyme that we inhibit, is an aminopeptidase in the endoplasmic reticulum, and its primary function is in peptide substrate cleavage prior to presentation on MHC1. So in any cell you will have proteins. These proteins will be degraded by the proteasome. Those peptide chains will then be transported into the ER, whereby a proportion of which will undergo cleavage, and then they'll subsequently be loaded onto MHC and presented to the immune system. So in the context of autoimmunity, the same happens for those autoantigens as does normal peptides. So they undergo some ERAP1 dependent cleavage, and they're in particular in axial spondyloarthritis, they are presented on B2705 to these autoreactive T cells. So that's what happens under functional ERAP1 conditions, and then our approach is really to turn ERAP off. And by turning ERAP off, you prevent this peptide substrate cleavage, and you block the presentation by changing the antigen repertoire at the cell surface. So you ablate this autoreactive T cell response. So axial spondyloarthritis is a predominantly CD8 T cell driven disease. And really what this schematic depicts here is just the lead hypothesis around the early event of this cross recognition of self-autoantigens. So there is an initial microbial event, normally an infection in these patients. This microbial peptide is presented on HLA-B27. It is then cross-recognized as a self-antigen by T cells in these patients, and this is where really autoimmunity starts. And I think just to highlight here, there's some really compelling genetics. So if you are an individual that carries a particular variant in ERAP1, and this particular variant is a hypermorphic variant, so you still have the ERAP1 enzyme, but it's just hypermorphic, so it's inactive. You are significantly protected against developing axial spondyloarthritis, and the association there is about 10 to the minus 41. So not only is that the case with ERAP1, but there is also an epistatic relationship with HLA-B 2705 in these individuals, and that association is 10 to the minus 27. So if you have a hypermorphic ERAP1, in those patients that are HLA-B 2705 negative, you are incredibly protected from this disease. So we really wanted to understand where the link was between ERAP1, the peptide presentation of these autoantigens in these HLA-B 2705 individuals. And to sort of tackle this from an exploratory perspective, we looked into 3 model systems using immunopeptidomics. So 3 model systems, PBMCs, where all of the PBMC donors had endogenous expression of HLA-B 2705, SW1353 cells, where we had to transduce in HLA-B 2705 allele, and then HT1080 cells, which do have endogenous HLA-B 2705, but it wasn't quite to the level we needed that, so we also transduced that in as well. And then we treat all of these cells with our inhibitor and generate pellets that we send to Biognosys, and then perform DIA-based immunopeptidomics. And all of these are searched in Spectronaut. So I think just on the right-hand side, just to go over this briefly, and I guess really just to say top graph on the right, I broke the axis just because we have 3 independent PBMCs where only the HLA-B 2705 is shared, so we naturally get many more peptide IDs there, so we get about 41,000. But in the SW1353 lines, we're getting about 22,000 peptides and about 14,000 in the HT1080. And then the bottom right really just illustrates how many of those are relevant to the model system we're studying. So we have about 9,000 HLA-B 2705 peptides to study in the PBMCs, 6,000 in SW1353, and about 3.5 in HT1080. So we started to try and characterize why ERAP1 inhibition may play out as a therapeutic in abrogating presentation of these autoantigens. And really just starting on the left-hand side here for these 3 volcano plots, what is very clear to see and we know is a very typical biomarker of engaging ERAP1 as a target. Because you are inhibiting the cleavage of these peptide substrates in the ER, you drive the presentation of longer peptides at the cell surface. So you drive presentation of proportionally longer peptides, and that is what is depicted here. So what you're looking at in these volcano plots, on the far left of these 7 to 9 mers that are mainly illustrated in purple, so you're mainly down-regulating 7 to 9 mers, and you're predominantly up-regulating these 10 to 14 mer peptides. And then the right-hand side really just depicts that in a length frequency plot. So if you look at the length distribution of those up versus down-regulated peptides, you can see the purple bar is what you would typically expect to be a very normal MHC1 profile, but you can see when you treat with a drug, you decrease those 9 mers and increase the frequency of longer peptides. And then this effect really is incredibly pronounced on HLA-B 2705. So the top graph being all alleles in that particular cell line, and the bottom being the effect of that length frequency on B2705 itself. So we have a really large effect on 9 mers here, and we really drive the presentation of longer peptides on this allele. So we thought that was maybe one piece of the puzzle. So maybe B2705 as an allele is just incredibly permissive to presenting these longer peptides. And we'd done some in silico work to try and see if that was the case. So we took these up and down regulated peptides on this allele, and we just looked with NetMHC pan to predict their binding affinity. And I know there's obviously a lot of caveats with that approach, but we were really looking for a hypothesis lead to go and validate further. But what you can see quite interestingly is that on the right-hand side, if you follow those purple bars from 10, 11, 12, 13, and 14 mers, you can see that they are actually all predicted to be stronger binders relative to any of those other alleles in that cell line. So there is something about B2705 that we think that will just readily accommodate these longer alleles, but that's not the entirety of the story here. So if you then look at the motifs of those differentially expressed peptides, so the top left graph here being those down-regulated 9 mers, you can see there's quite a strong restriction for arginine and glutamine at the P2 position. If you then take the upregulated peptides for the 10 mers and the 11 mers, and this also plays out for 12, 13, and 14, but I didn't think fitting 5 graphs onto one slide was really going to be that constructive. You can see there is a really strong preferential retention of this arginine at P2. So not only are these peptides just longer and just stochastically random, you have to retain this P2 position as well for presentation. So we'd map this back then on the right-hand side to the context of these autoantigens. So there is a paper out in Nature that had used a yeast display system, and they'd screened these HLA-B 27O peptides for autoreactivity against axial spondyloarthritis patients' TCRs. And they'd come up with this list of 9 putative autoantigens. And you can see that all of those 9 mers, those putative autoantigens, will have that arginine or that glutamine at P2. So then if you think about this in the context of what our drug is doing, so in the context of ERAP1 inhibition, you will extend the N-terminus of these peptides. And if you take the N-terminal extension of these peptides, what actually happens is you remove that P2 preference completely. So we think this is not just a length thing, but it's a length and loss of specificity, which I guess makes sense from a binding perspective. So our hypothesis here really was that N-terminal extension of these autoantigens would just ablate their binding to HLA-B 2705. And this is the sort of hypothesis that we generated that we then went on to further test experimentally. So we show in a Mauditov system that you can get very, very potent inhibition of mature autoantigen production for a number of these autoantigens. So we didn't test all of them, but these are 3 that we tested, and you get picomolar affinity against these antigens with our inhibitor. And then interestingly, to sort of prove this hypothesis about N-terminal extension and loss of binding specificity, we take a mature autoantigen here with this arginine at P2 and test it in a purified HLA-B 2705 binding assay, where you're essentially asking relative to a labeled reference peptide, how much can you displace based on binding affinity? And what is really interesting to see is that you take the 9-mer, it has high affinity for B2705. You then extend, so you generate a synthetic peptide with an N-terminal extension, just one single amino acid, and you decrease the binding affinity by 60-fold. You do 2 amino acids, and you decrease it by 120-fold, and 3 will get you to a 170-fold decrease in binding affinity for this particular autoantigen. And then I think just as a positive control here to show how important that P2 position is on this particular allele, you can take the 9-mer and just replace the P2 arginine with an alanine, and you also get the same effect as extending it by +2. So we've really shown here, I think, that by N-terminally extending these peptides, they're just not making it, in the world of competitive dynamics in the endoplasmic reticulum, they're just not making it back onto the allele. And that's what hopefully we've illustrated here as well. So we now take a selection of these pathogenic TCRs, so these TCRs that have been shown in the paper to be cross-reactive with these autoantigens. And we first validated that cross-reactivity existed, so we made a JERCAT-based system that expressed these TCRs, and we also overexpressed this autoantigen as a 14-mer minigene. So a 14-mer minigene that was tagged to go directly into the endoplasmic reticulum. So what you can see, and what is illustrated by green here, is just that all of these TCRs recognize the mature autoantigen. So we know that is a fact. And then if you, again on the right-hand side, if you take this system and you treat these cells in this co-culture with 715, you can decrease the activity of all 4 of these TCRs against this autopeptide. So this is, again, just to reiterate, this is 14-mer into the ER, under normal functional conditions, is cleaved by ERAP to produce that mature 9-mer autoantigen, obviously then cross-reacts with T cells, and then you inhibit that process and you get no presentation at the cell surface, and you can ablate this T cell response. And not only can we do that, we can do that in a dose-dependent manner as well. So the graph on the bottom here just shows that we can do a titration of our drug and get a really nice dose response. So I think just to wrap all of that up, I guess quite a lot of different data. We used immunopeptidomics to define a hypothesis and do some exploratory work to try and understand why, based on the genetics of this disease, ERAP1 inhibition could play therapeutically in this particular condition. And based on that hypothesis, we went on to do some enzymology and binding assays, and we show that ERAP1 inhibition not only prevents the mature autoantigen production, but it does so by the N-terminal extension of that peptide ablating binding to B2705. And then the final piece of the story was really the T cell activation. So if you create a system where T cells recognize this peptide, you can completely block that recognition by inhibiting ERAP1 in this system. And just to say that we opened a phase 1/2 clinical trial for this disease earlier in the year for HLA-B 2705 positive patients. And I think just with that, I'll wrap up and say that this is by no means all me. It's absolutely a team sport to develop drugs. And I think just to thank Biognosys again, but also thank all of the people in this picture who help and contribute to this work.